Hi everyone, welcome back to a video series on cell division. This is Biotan, the host of the video. In our previous video, we learned three things. We learned about what diploid and haploid means. Diploid means that we're talking about two sets of chromosomes, whereas haploid refers to one set of chromosomes. So that means to say we have two of every chromosome within our cells, because most of our cells are diploid in nature. A pair of these chromosomes we refer to as homologous chromosomes. Homo means the same. So when we look at homologous chromosomes, they are largely the same, but they are different in certain ways. They differ because they have different forms of the same genes. We refer to different forms of the same genes as alleles, and each allele results in a slightly different form of the same protein. So in today's video, I will be comparing and contrasting two forms of nuclear division, mitosis and meiosis. But before we can compare the two, let us do a quick recap on what we already know, the cell cycle. The cell cycle as we know it consists of G1 phase. G1 is the phase where the cell grows. S phase is where DNA replication occurs, and G2 is where the cell grows even further. Together, collectively, we refer to this entire phase as interphase. Essentially, the cell prepares during interphase to enter mitosis. We want to produce double of everything that we have. Mitosis consists of four phases. We have prophase, metaphase, anaphase, and telophase. Together, we call this mitosis. Mitosis is a form of nuclear division. In other words, division of the nucleus. After that, the cell divides into two daughter cells in a process called cytokinesis. And cytokinesis is a form of cell division. So just to make a clear distinction, there's nuclear division, which is mitosis, and then there's cell division, and that's when cytokinesis occurs. Let's look at each phase a little bit deeper. Mitosis begins first with prophase. And at prophase, we see that the threads of chromatin fibers begin to condense to form what we call metaphase chromosomes. And metaphase chromosomes are much more condensed, and you can start to see them under the light microscope. And these metaphase chromosomes look kind of like butterflies. That's because they consist of two sister chromatids. Here is one sister chromatid, and then here's the other sister chromatid of this particular chromosome. Why are there two sister chromatids? Just because the cell has undergone the S phase, which is DNA replication. We also find that in prophase, the nuclear envelope begins to disintegrate. And we start to see the beginnings of mitotic spindle extending out from these centrosomes. And some of these mitotic spindles, as they extend out, they will start to attach to the centromere regions of chromosomes. So now let's zoom in and have a look. Here I have a chromosome on the right hand side with two sister chromatids, sister chromatid 1 and sister chromatid 2. You find that in the middle, this is where the centromere region is. At the same time, you'll find that there are some proteins at the centromere region. We refer to them as kinetochore proteins. This is where you'd find all the kinetochore microtubules attaching to the kinetochore. If I were to look at the cell, you'll find that the kinetochore microtubules will come out from the centrosome from both of the opposite poles, and they will attach to the same centromere. And a kind of tug of war starts to occur. In this epic tug of war, the kinetochore microtubules will adjust their lengths until the tension on both sides become somewhat similar, and at which point you'll find that the chromosomes get pulled to the middle of the cell. We call this midline the metaphase plate, and this is where the chromosomes line up singly. Here we have entered metaphase. In metaphase, all the chromosomes line up singly, as you can see here. Homologous chromosomes still exist, and you can find them lined up anywhere along the line. For example, here is one particular chromosome, and its homologous chromosome is somewhere at the bottom. Here's another chromosome, and you find its homologous chromosome elsewhere. So just bear in mind the homologous chromosomes still exist. Once all the metaphase chromosomes have lined up, the cell then undergoes anaphase. This is where the centromere divides, and we find that the sister chromatids are pulled to opposite poles. And they are pulled centromere first, resulting in the characteristic V shapes. Once separated, we refer to each sister chromatid as daughter chromosomes. At each pole, when the daughter chromosomes have been pulled to the opposite poles, we find that the DNA begins to decondense and uncoil, and the nuclear envelope begins to reform. At the same time, cytokinesis begins to occur. You start to see a cleavage furrow form, and eventually these cleavage furrows will deepen and deepen until eventually the cell divides into two. That deepening of the cleavage furrow, we call that entire process, cytokinesis. And this is what we get at the end of the day. We get two what we call daughter cells. These daughter cells contain the same diploid number of chromosomes as the original cell. Here we see that there are two of every chromosome. You find that the homologous pairs of chromosomes still exist, right? And this is a stark comparison to what we saw somewhere at the beginning. This is the chromosomes that you saw at the very beginning of prophase, where the chromosomes exist as kind of like butterflies, right? But because at anaphase, the sister chromatids are pulled apart, what we get as a result is this, just single daughter chromosomes in each cell, right? But a diploid number of daughter chromosomes. So now let's compare and contrast this process with meiosis. Here we have meiosis, the cell cycle of meiosis, and we can see that it consists of many, many more phases. When we look at meiosis, the cell undergoes 2 rounds of nuclear division. Here's the first round of nuclear division, and then there's the second round of nuclear division. We find that while we can still carry out prophase through to telophase, we do this process 2 times. Prophase 1 all the way to telophase 1, and then prophase 2 all the way to telophase 2. The mechanisms involved are largely the same in each phase, so I will instead focus on the differences and compare them to mitosis. Okay, we call this entire process meiosis. So meiosis consists of 2 nuclear divisions. Now, we will look at meiosis as a whole. Okay, give me a moment as I make all these visible. Okay, let's go into meiosis. Meiosis begins similarly with prophase. We call this first prophase, prophase 1. You also find in prophase 1 that the chromosomes the chromatin fibers begin to coil and condense to form distinct metaphase chromosomes. But we find the first difference here. The first difference is that the homologous chromosomes pair up during prophase 1. What do I mean by that? So if this is one chromosome here, it will find its homologous pair, and then it will pair up with its homologous pair. We call this pairing up process of homologous chromosomes as synapsis. So synapsis is a process. We call this pairing up process synapsis. So another way to say this is that synapsis occurs during prophase 1, where all the homologous chromosomes pair up. I've simplified this diagram so we see that 1, 2, 3, 4 pairs of homologous chromosomes, right? 4 pairs of homologous chromosomes here. The implications of this is that as the kinetochore microtubules attach to the pair of homologous chromosomes, they will then bring these pairs of homologous chromosomes to the metaphase plate 2. So during metaphase 1, we find that rather than the chromosomes lining up singly, we find that pairs of homologous chromosomes, we have 4 pairs here, so let me draw them all in. The homologous chromosomes pair up and line up at the metaphase plate, as opposed to in metaphase or mitosis, they line up singly. All the chromosomes line up singly. And the implications of this is that, the kinetochore microtubules will still attach, but when the kinetochore microtubules begin to shorten, what you find is that rather than separating the sister chromatids apart to opposite poles, we instead separate the homologous chromosomes to opposite poles of the cell. And as a result, at telophase 1, we find that each chromosome still consists of sister chromatids joined together at the centromere. At this stage, the chromosomes may uncoil and they may form chromatin threads again, and the nuclear envelope may or may not reform. It really depends. So, however, cytokinesis will definitely still take place. At the end of cytokinesis, we will end up with 2 daughter cells. However, this time, each daughter cells contain a haploid number of chromosomes, or just one set of chromosomes, whereas the daughter cells of mitosis contain a diploid number of chromosomes. Another way to look at it is that each daughter cell now contains just one of each pair of homologous chromosomes. The other homologous chromosome of the pair is found in the other cell. Next, we then go to prophase 2. We carry on these 2 cells to prophase 2. So this is the first cell, and then we have the second cell. We take them on to prophase 2. In prophase 2 all the way to metaphase 2 to anaphase 2 to telophase 2, you find that the mechanisms involved is very similar to what you find in mitosis. But we begin instead with a haploid number of chromosomes in each daughter cell. So for example, what do I mean by it's quite similar to mitosis? You find that the chromosomes at metaphase also lined up singly. And at anaphase, you find that the sister chromatids are pulled apart. And when they are pulled apart, we refer to them later on as daughter chromosomes. And then at telophase, the daughter chromosomes uncoil to form chromatin threads again. Nuclear envelope reforms, and cytokinesis occurs. This time, we end up with 4 daughter cells in total, right? Because we started off with 2 daughter cells above over here, make it go through cell division. Here, we end up with 4 daughter cells, each with a haploid number of chromosomes. So you'll find a haploid number of chromosomes in each cell. Okay, with that, let us summarize some of the key differences between mitosis and meiosis. Let's zoom out. Okay, so the key difference between mitosis and meiosis is this. Number 1, homologous chromosomes undergo synapsis in prophase 1. You find that in synapsis, the homologous chromosomes pair up. Compared to prophase or mitosis, homologous chromosomes do not pair up. Difference number 2, homologous chromosomes line up in pairs along the metaphase plate in metaphase 1 of meiosis, while chromosomes line up singly in metaphase at the metaphase plate in metaphase of mitosis. Number 3, homologous chromosomes are pulled to opposite ends of the poles in anaphase 1 of meiosis, whereas in anaphase of mitosis, it is the sister chromatids that are pulled to opposite ends of the poles. Number 4, at the end of meiosis, we get 4 daughter cells that are haploid. Whereas in mitosis, we get 2 daughter cells, each diploid in nature. If you want to look at similarities, actually, the entire process of mitosis is quite similar to prophase 1 all the way to telophase prophase 2 all the way to telophase 2, except that each cell in prophase 2 is working with a haploid number of chromosomes. Now this was a long video. I thank you for following through to the end. I'll see you in the next video where we'll talk a little bit more about the amount of DNA and sets of chromosomes.